Hello, medical students, residents, and fellows, and my dearest colleagues and consultants and cardiologists uh, all over the world. I'm here able to talk to you via this wonderful video that I'm, presenting through MuniMeterHealth.com, which is our website. I'm Dr. Jaitly from New York, and I'd like to discuss today in this particular video section something which is so unique to a cardiac cell. And let's try to understand what the structure and the function of that cell is. The cardiac cell is called cardiomyocyte. Obviously, just like any other cell, it is extremely unique, and therefore it has its own function. Now, what does a cardiac cell do? Well, contract and relax, depending upon what the stimulus is, etc., etc. Just like any other cell, for instance, you have retinal cells behind your eyes, which are giving you the ability to see and appreciate the colors around within the surroundings, or on your fabrics, or on your furniture. Likewise, you have skin cells. They allow you to make you feel, get the feeling, get the touch, etc. And similarly, you have cells within the gastric mucosa lining, and they have only one function: to produce hormones and enzymes and etc. to ability to facilitate the digestion. Similarly, the cardiac cell has its own function. So the basic mechanics of the cell, the basic engineering, if you will, is very similar. Like you have a membrane, you have a nucleus, you have a what you call SR, the sarcoplasmic reticulum. I'm sure some of you are already familiar with these terms. Those are called organelles. So anything within the cell is considered as organelle, and each of these cells, they line up to form the tissue. Now this is the heart. Obviously, this is a macroscopic view of the heart. But when you look at individual myofibers, say if you were to take a section of the heart, you'll see a lot of striated myofibers. So the myofibers have striations, and this is to the naked eye. So you'll see a lot of fibers which are attached to one another, and they are branching off, etc., on this left panel, as you see. Now, if you were to look under light microscopy or a light microscope, you will see each of these myofibers will be lined by cardiomyocytes, and one myocyte is almost a dimension of 10 by 20 in the atrial microns. We're talking about microns, and roughly about 70 to 150. They're much longer in the ventricles, so 70 to 150 microns in the ventricles. I mean, they're extremely microscopic, as you see. So the light microscopy will have cylindrical cells, more or less cylindrical cells, and they're nucleated. Each myocyte normally has one nucleus. Sometimes they have multiple nuclei, but one nucleus. And a nucleus is obviously containing the entire genetic material. So a nucleus contains the chromatin. It contains all the genetic stuff, what determines the contractility, your longevity, and everything else. So this is, or it also defines during the process of an illness how it handles the hypertrophy. If the hypertension was to develop, would the cells start producing more proteins to develop more cells, so that the myocardium actually here becomes thicker, and which is commonly seen in hypertension. So there's a lot of genetic stuff going on within the nucleus. So, but the point here I'm making is very similar to any other cell. The entire engineering within the cell is quite similar. For instance, you have the mitochondria, which is the energy producing zone, so if you will. And there are numerous mitochondria scattered throughout the cell, and you can tell that this cell is energetic. If you don't see too much of mitochondria, then you know that cell is suffering from energy. It's going to be a little sluggish cell. So that shows that patient is having heart failure, or may develop heart failure, is not doing too well, is not nutritionally sufficient, or there's something else going on. There's a virus that's affecting, and therefore it's causing myocarditis and what have you. So there's an energy lacking. That's an energy lacking cell when you have less of mitochondria. Obviously, all of this is interspersed within the cytoplasm. The cytoplasm is called, and the fluid is called the cytosol. But then the cell membrane is called sarcolemma, and that's a very active membrane of a myocyte. It's not like just like a covering. Obviously, it protects one cell with the other, but you can just picture yourself that there are numerous cells lining all of this, making a huge myofiber, and all these collections of myofibers, tons and millions of myofibers form a heart muscle. Get it? So myofiber, the heart muscle, the next step is myofiber, the smaller step is myocytes and series of myocytes. So now we are inside the cell and we are looking at the organelles, every single organelle. What have we seen so far? Mitochondria, cytoplasm. Sarcoplasmic reticulum is something which is like actually an invagination. I tried to show you here. It invaginates within the cell from the sarcolemma, which is your membrane. So when it invaginates, obviously it creates a huge surface area. Imagine you're still holding a pencil, for instance, and that pencil has some crevices, for instance. You create some crevices, by small little things. And if you create some crevices within the pencil, you'll have increased surface area of that wooden pencil. Similarly here, the cell invaginates inside of the sarcoplasma, has a huge surface area within the cell, you can imagine. And those sarcoplasmas, they will have calcium trigger zones. Now, we know the sarcolemma also has an active calcium pump, which requires energy, and energy is ATP, if you will. Just like car runs on fuel, and the fuel there is gasoline. The cell runs on fuel or energy, and that is ATP. And the energy is all stored within the mitochondria here within the cell. So having said that, the sarcoplasma, for instance, now the calcium is brought in by energy producing pump, energy using pump, and the calcium pump brings it in. One will see that the heart muscle will contract or relax depending upon the uptake or the release of the calcium. Very interesting stuff. Now, just pay attention here. Within the myocyte, what makes it contract are these proteins, and these proteins are called myofibrils. Now, these myofibrils are again microscopic. Now we're getting electron microscopic here. So when you go inside the cell, within the cell, what you've seen is a nucleus and all these organelles we just talked about. That's on light microscopy. Once you get into the electron microscopy structure, one starts seeing all these filaments, and these filaments are composing the myofibrils. So there are tons and tons of myofibrils lining the cell within one cell, and those cells, and each cell has its myofibril, and each of these myofibrils are composed of 3 types of filaments. One is an actin filament, which is a thin filament, and you could see that's anchored here to the Z line. We call it a Z line here. Myosin are the thick filaments, and they are starting from the middle of the Z line, middle of the Z zone, if you will, and they extend in this direction. Now, at the end of the myosin, there is another protein that's attaching to the Z line on the other end, and that's called the titin protein. This titin protein has only been recently discovered. We have not known it for, we have known it only really for less than 10 years. So it's a very, very interesting aspect. We're still growing in our knowledge within the electron microscopic world of the cardiomyocyte, just so that you know. But the mechanics and the engineering is amazing. It's just phenomenal. Why? Because these actins and myosins, as you see, they will move, or they'll slide over each other, if I will. They slide over each other in this direction. They slide over in each direction. And there are numerous hundreds and hundreds of millions of these such myofibrils within the cell. And you can imagine microscopic millions of cells within to form the myofiber. And all of them, they work in synergy. And when they work in concert, just like your philharmonic, whatever, the orchestra, one player does not march on the band, and the whole thing just doesn't sound like music. It sounds like noise. And similarly here. So the cells did not contract and relax in synergy. That heart muscle will not function normally. So in order for it to function normally, let's understand that the actin and myosins of all individual myofibrils within the cell, they must work in synergy. Very, very interesting. So, and we'll delve on these actin myosin titans on another video again, but because that requires a little more intricate stuff. So thank you for your attention. I think this was a short video on cardiomyocyte. Hope you got a grasp. The the naked eye sees the myofibers. Myofibers are consisting of multiple hundreds of millions of myocytes, and within the myocytes there are different organelles, and within the organelles, of course, the heart cell is uniquely qualified to have myofibrils, and myofibrils are obviously consisting of various proteins, and these proteins, they slide over each other. They are called actin and myosines, and the recently discovered titin, which is also anchoring the myosin filaments to the Z lines. Okay, thank you for your attention, and looking forward to seeing you on my subsequent presentation, which will be on sarcomere, because sarcomere is this one unit between the two Z lines is the sarcomere, and we'll talk about that in the next one. Okay, stay tuned. Keep watching MuniMeterHealth.com on YouTube and Facebook. This is Dr. Jaitly from New York. Thanks for your attention.